interested in this. We are going to start right now, and we have a kind of political panel as well. And it is also addressing the big chunk of dystopia behind the thin layer of utopia in a certain way, in both cases. It's addressing the relationship between the promises of greenness, greenwashing, greening the world, alternative energy production, and carbon sequestration. So a lot of issues on this panel, and the first duo, so to say, that we have on this panel is artist Tagny, and along with Dana Kirk, assistant professor in Biosystems and Agricultural Engineering here. So the first panel is about wastelands, and wastelands is a project with which Tagny has been invited here to the British Artisans Resident program as a winning candidate. We had something like 50 applications, and the jury was anonymous in finding Tagny's project the most powerful. And Tagny is an artist and researcher from Canada, from Montreal, and having been coming into countries for several times to work on a kind of future, a near future, where we will source our own energy from waste materials after this disastrous period of fossil fuel combustion. So, may you go ahead to explain what your wasteland is like?
Okay, sure. Can everybody hear me? Let's just see if I can. Okay. It's 2,518. Microbes like E. coli, phages, and viruses are found in human and living excrement, and they are acknowledged as an extension of our kinship, not as biotech workhorses or germs to be standardized and controlled. The social order has transversed all carbon life forms. Technological research develops from an anachronistic sense of tool making and material use, ranging from prehistoric human history to an unknown future. Paleolithic discoveries of technologies coming from rock and stone become mainstream. Mega military complex and fossil fuel-driven capitalist industries no longer exist. Humans recuperate their feces towards the production of light and heat. Girls are the leaders in STEM, society, technology, engineering, and math. Girls are leaders in political leadership. The social order has changed. It now follows poly relations, not observing hierarchical power structures. So this project that I'm going to talk about is a new work, and it's been created specifically through the residency here. I've been to the campus 3 times, and I've been developing a series of soft biological art sculptures based on this idea of living 500 years in the future in a world where fossil fuels are no longer used for energy production, and instead human feces are used as material for energy, a natural energy source to heat different types of shelters and different types of living materials. So to do this, of course, I have worked in an aesthetic that really looks at recuperating contemporary types of materials that are either biodegradable or sourced from plant-based materials. So in the sculptures that we see here, it's a sort of a sneak preview of what you're going to see at the reception this evening if you do come to the gallery. There are 2 sculptural prototypes that are showing you a biogas generator system that is fed with various types of digestate, and digestate is coming from cow manure and also food waste from the MSU campus itself, and it's being used to feed. It's being used to produce methane, a very small amount of methane, and hypothetically, this methane production can fuel light sources and different types of heat sources. But in these sculptures, what you're seeing is a reading. There's a monitor, the water monitor there shows the methane production coming out of the one sculpture on the bottom there. There'll be there's a meter reading that will reflect how much natural gas is actually produced. And on the top, and I'm going to go into more detail, but the second sculpture on the top is another custom-made biogas digester, and it is linked to a balloon that absorbs all the methane production. So these are very small-scale kind of prototypes. They're meant to be low-tech. In fact, anyone could build one of these. It's based on a DIY prototype that can be found on the internet. It's actually a high school level science experiment. Okay, so what's interesting about this sculptural work is that while it is low tech, it is also very much invested in the high tech rhetoric around new biogas generation and the potential of so-called green technologies and green gas. So the project was really looking at how to upscale and recuperate a lot of used science technologies. So in this case, used science beakers and different kinds of scientific equipment was purchased secondhand from, there's a place here on campus that sells used science equipment. So again, low cost, easy to find, and then the glassware was recombined based on a design that I had made, a very simple design for the biogas generators. And so you can see there's a number of them here, but there's the long one and one of the other glass sculptures there are used for the show that you'll see tonight. In order to troubleshoot these biogas generators, or digesters as they're called, I worked with Dana Kirk, who's here with me, and I worked in his lab, and he and his lab team were very generous to get me the digestate from campus in order to run the test to see how much methane was produced from these very small prototypes. And here we're troubleshooting the setup just from a couple weeks ago. You can see the digestate that we used, like I said, is sourced specifically from MSU, and it's taken and then, let me just show you here, we put it into the digester there, and then we ran it through the experiment where we had it in a heat room, and the hotter the digestate is in these digesters, the more methane production we're going to get. And this is very much a common scientific setup that is used in Dana Kirk's lab for his own research. As well, because I couldn't be here so often, and also with an interest in trying to work locally, I spent a lot of my time developing this in my own lab in Montreal as well, before coming here to do more of the troubleshooting with Dana in his lab. So I was able to run the methane accounting in Montreal with a little Arduino methane sensor. So again, really low tech, very low cost types of technology. So I'm very much invested in this idea of how can we engage in these so-called green technologies in a way that is more accessible to a larger group of people, both scientists and non-scientists. While I was in the lab in Montreal, I also was developing and working with 2 other artists, Courtney Brooks and Whitefeather Hunter, in developing a new mushroom-based material, so a bioplastic, in a way to think about how to host the biodigesters in a material that is post-fossil fuel, kind of era-driven material. And this material that we came up with is a kombucha. It's from mushroom. And it really is Courtney Brooks and Whitefeather Hunter that completed the protocol for that. That's their research. But we all worked together to try to find a formula and a protocol in order to make this new material rigid enough to actually be used as a container and a host for the digesters. And so that took 6 months of work. We grew them, the kombucha material, in big vats, big pools. And then what we did was we then, after we'd grown the kombucha for the bags, I had Whitefeather and I worked together to develop this design in order to fit the one biogas digester there, based and modeled on a very old, ancient type of stone bag that is found actually all over the world, many, many different forms of different types of cultures. And this kind of very ancient prehistoric stone represents a kind of coming to reunify the living and the non-living material existence in the world, and the sense of cosmos from a very early time. So this project is also looking at how we can invest in technologies across human history. So instead of looking at the narrative of technological progress as moving forward, we kind of look at going backwards and forwards and in the future and jumping around through history to find different kinds of technologies and to see how we can reinvest in this narrative that we have today around green technology. And the second prototype here is again made with the same bioplastic, and it's modeled after a Paleolithic carrying case. So again, this is a type of wearable now that is referencing prehistoric narratives and use of different types of technologies, and kind of bringing them all together and conflating our notion of technology as being this kind of progressive narrative that will save the future. Instead, it's a looking back and kind of thinking about different types of materials, and also a different way of acknowledging potential technologies that could be reabsorbed and repurposed and upscaled today. And so having developed the new material and creating the new bags within the custom-made digesters, this photograph has come out of the project, and it's really kind of a way of expressing the use of this so-called new technology in a different way. Here we see the 2 girls are holding the carrying bags with the biodigesters, and they are kind of plugged in, so there's a piping that goes, you can't quite see it, but it goes around their body, and it's suggesting that it's actually coming from the back, so that there's a recycling of human feces to actually be able to light up a bag and also act as a heat source. And in this case, the body also is a heat source that can activate the biodigestate that's in the bags and in the generators. So it's really a kind of an investigation into this, working through different imagery and histories around natural gas technologies, and imagining, again, how can we really rethink of this notion of the future by kind of looking at history through a very different lens. And there are many, many people who've worked on this project, so I definitely want to acknowledge the Bridge Artist Residency Program, and of course Dana Kirk and the ADRAC labs that had a big part of this as well, and then also the Speculative Life Biolab in Montreal, and my other collaborators, Whitefeather Hunter and Courtney Brooks. So on that note, I think I'll turn over to Dana.
Yeah, so a little bit about, I guess, the work that we do here at Michigan State. We have a laboratory and digesters on campus, and the work is focused really on, again, very similar to this, repurposing organic waste. We're very good at recycling plastics and metal and cardboard and paper, but when you look across the United States and across the world, organic waste, food waste, human waste, animal waste, are we as efficient as we could be? Are we as effective at getting the most opportunity out of those waste products? Probably not. And so using technology like anaerobic digestion, which is not at all a new technology, it's been used for thousands of years across civilization to address waste and human health through killing or reducing pathogens and producing methane for lighting and heating purposes. So it's not at all a new technology, and the microorganisms that we're harnessing, the hundreds of different microorganisms have been around for millennia. So we're doing it, in a mechanical way, a very mechanized way. And in fact, if you see our digesters here on campus, they're full of sensors and lights and all kinds of buzzing things, and they'll alert my phone if they're not very happy at any time of the day. And so, it is really an engineered extension of our intestine or the stomach of a cow. And what we're doing is taking and continuing that degradation of organic material and actually creating a renewable value there. And so the focus right now across the United States, North America, is to take cow manure or human waste and convert it into actual pipeline quality natural gas or methane and use it to fuel vehicles. And so that is the real world application that's driving the research that we do, that's driving investment and advancement. The goal for California is to have 10% of their vehicle fleet running off of renewable natural gas from waste in the next 20 years. And so it's a big motivation to address this issue of waste, climate change, emissions, odor, but also just recover, reuse, and repurpose things that we would typically throw away and not think of. I'm not sure that's the debate you were looking for.
Oh, I think so, yes. Still, you were mentioning yesterday, well, we are now in a stage that this discussion has become so exciting that you are possibly handing in a research application together. I mean, we talked about different modes of research in arts and science, so that artistic research is being understood as something completely different normally than scientific research, but in how far the motivations here would overlap, both to have the scope of making a kind of significant progress in knowledge production, but also in a cultural interpretation and towards the criticality that artists are very strongly looking at. So in how far this goes together, a kind of both utilitarian and non-utilitarian approach to that?
That's a great question. I mean, that's always my approach is not for it to be necessarily a utilitarian object. I mean, clearly it functions, but my intention is never for it to be a functional object that I'm going to put into commerce. Whereas with Dana, that is the intention of the research. But yet we still have this overlap and interest in thinking about how to also scale up the project a little bit. We were talking about potentially moving this into a more of a human scale, where we could be thinking about working on a garburator system, which is what Dana's been thinking about, something that would allow every person to access this type of mechanism or digester in their home to be able to generate their own energy. But for my interests, I would like to see something like that function, but I want to see it as a way of scaling up the work that I've already done in this residency, and kind of push it further so that I'm challenging people's sense of aseptic technique, really trying to work with feces as a material, as an artistic type of material again, and questioning our notions of hygiene in a home environment or a lab environment. And thinking about bacteria interactions with feces matters, and also with viruses. It's another interest of mine is to work with phages and bacteria, of course, that you find very often in this kind of digestate. And so one thing I didn't talk about was that work with the phages, which will work as a kind of an off switch to the bacteria, especially E. coli, that helps with the methane production. So I'm interested in that, in the kind of relationship between bacteria and phages and humans, and how that might be scaled up to be experienced within a gallery environment.
Your paper that you gave in Copenhagen recently was called S-Statics.
Yes.
So, I mean, I guess it is not going to be the name of your funding application, but in how far does the language and the criticality and also the subversion that goes into Tagny's research, I mean, it was obvious that it's bringing up gender issues, power relationship, and so on, issues of petrofascism, and so on. And how far do you deal with that in the context of then making sense of this, enriching your own science? And how far do you feel that it triggers something that is pointing to new ways when you are thinking about your own research?
Yeah, it is. I mean, it's we know that we're shifting our energy sources. And one of the big challenges that we face with how we're doing it today is how do we store energy? If we do solar, we have no way to store it. If we do wind, we have power when the wind blows, but we don't have power today. The leaves aren't even shaking on the tree. And so if we were just relying on wind, we would be out of luck today. And so if we can actually control the biological space, we know that that is probably the most efficient and inexpensive battery or energy storage device that we might have. And so being able to turn things on and turn things off at our desire or for purpose has some real opportunity and could serve if we want to power our house off of human waste and animal waste and food waste. When you think about all the stuff that's carbon-based that goes out of our house, there's a real opportunity for energy there.
And I think that, I mean, we've had discussions about the aesthetic and the way that I like to kind of confront people and their fear around messiness and the hygiene. And I think that just working with the material in the science laboratory also becomes much more of a mainstream, like everyday experience, so that kind of aesthetic relationship is there in the lab, and that there's not as much fear or concern about that in a scientific research context, because I mean, Dana's working with like shit every day. It's like, that's what it is. So, you know, it's actually refreshing for me to go into a lab like that compared to all the kind of tissue cultured labs I've been in, biosafety level 2, everything's so clean and aseptic techniques everywhere. But with Dana's lab, it's like you smell it, you know, it's like on the farm, it's everywhere.
I did shower before I came and changed clothes.
Yeah, so it's you know, it's a different kind of context.
So we are going to talk about smell later on in the last part of our section, and so there can be a kind of anticipation of Cecil Tola's project about a smellscape being developed about a specific neighborhood in Detroit. But before we go there, we have another research project we are talking about now, also involving a scientific department here, or many scientific departments here at MSU, which is also dealing in a certain way with green energy. Also with the ambiguity that comes up when you actually scratch the surface of a metaphor, that of greenness, for example, that greenness here unfolding in the materiality of chlorella algae that is boosted commercially as superfood, at the same time that we encounter the chlorella algae also in other circumstances, that is, for example, toxic algae blooms, but also in research that is being done here about CO2 sequestration. So it's very much about how the materiality is changing in order to context. And here we have a duo or trio with Heiko Hansen working together in France, in Le Havre, since some months, together with Helen Evans. So they are French, German, British art duo that reflects on the energy needs of contemporary life. They are visualizing social, industrial, and ecological paradoxes through installations, performances, very large-scale urban interventions, or very, very small domestic hacks. So very often associated with what are called man-made clouds, and how the energy in these clouds is being hidden away, or the energy production. And the repertoire here includes technological accidents, but also the measurement and the codes of measurement of ecological measuring, for example. And they came across, and this is probably the part of Heiko's interest for this area, because they have been working around the Lake Erie, and together precisely with Susie Liu, an associate professor in Biosystems and Agricultural Engineering here at Michigan State University. And for the first time, I remember when Heiko came, he will probably tell the story, he was very amazed also by, or Helen came for the first time, and now it's Heiko developing the project, this huge existence of this power plant that is really responsible for the energy production of the whole site. And to really think of, how can we get this CO2 neutral? And there's research to be done about this. And so we have a perfect match also of artists making research to find actually a department here on campus which is enriching their resource, and hopefully in turn researchers collaborating with the artists getting new idea about how to develop their own science. So we'll probably start with Heiko Hansen, who is there remotely connected. Heiko?
Hello.
Hi.
Hello. Hi, I hope everybody can hear. Hi.
So we have a first image on there on the green cloud.
Yes, I've got one here. Hello everybody. It's already dark here. It looks very bright and energetic over there. We have kind of 9 o'clock here in France. I would like to thank straight away everybody for having invited us, and to the residency program. Of course, MSU and the Broad Museum, Jens and Adam in particular, and especially Susie for her kind of enthusiasm really to engage. I think I'm back now. Yeah, so I think I'm back now. Okay. So for Susie, it's really enthusiasm to engage in this adventure over really like the last 2 years on and off through many different ways, like over Skype, and of course our experimentation and exchanging ideas on site. And I just want to kind of maybe extract a bit, what Jens already mentioned, and illustrate that also with some other works of us, which I think you can see, if I'm not wrong, we see right now one of our works on the screen, which is Nuage Vert. Is that right? Yes, green cloud. Yeah, I presume I can't hear anything. So this was really for us fantastic to really meet Susie, and it's quite surreal really that we're kind of working on the same project in some way. Having worked on really how to turn emissions green, and then Susie actually working on the same idea to turn emissions literally green. So I mean, we are kind of doing it by converting the emissions into a light sign, and Susie uses the CO2 in the emission to feed algae into a bioreactor. So it's kind of both of us greening emission, and I thought that's also maybe an interesting starting point of what could be a definition of maybe how different working methods are, if they're very, very close in some ways. Maybe we have some time to talk about that later as well in the discussion. Maybe we can go to the Erie Lake slide as the next, the second slide.
Well, you had the power plant before, but okay.
Yeah, that's the power plant. Sorry, that is actually the power plant. Is the TB Simon power plant in East Landing, where also right now the bioreactor of Susie is installed. And I think that's where you should be receiving right now the energy to run this presentation from. So let's plug it in there. So if we go to the next slide, maybe of Erie, Lake Erie.
Yep.
So I think one of the most interesting things really for us in this residency was that we learned the extreme ends really, which we associate with algae today. And Jens mentioned that already. On the one hand, this idea of a future magic nutrient and power resource, whereby I find it quite interesting that actually this idea of using algae, and particularly the chlorella strain of algae, is somehow also a kind of, I guess, in some ways a fantasy of the past. I mean, without knowing too much about it, as I think a concept which appeared after the Second World War as a future kind of vision to feed the masses. And so there are these two ends really, and it's always, of course, in new technologies like that there is a kind of potential for a great new product, and at the same time a dystopian future. So there's always a very interesting point in time. I have also maybe to say that, you know, often Helen and I presume we are kind of situated like every artist and somewhere between the past and the future. But it's true that we often really start from an image which is important to people today somehow, or maybe right now. So I think having kind of having these two extremes at hand, which is on the one hand a kind of magic future product, but also the current role algae can play, which is a green plague really, which is quite common in not only in the region of the Great Lakes, but of course also in other parts of the world. For example, I've been the last days in the Bretagne, and it's also the same in France, that we have every year the reoccurring phenomena of algae blooms. But of course, if it is a particular phenomena which appears every year very heavily in Lake Erie. And as Jens mentioned already, one of the chlorella strain algae, which also Susie uses in her lab, is actually coming from Lake Erie. I had to think when I was just preparing this a bit, after Jens asked me a couple of days to go for slides, and I was looking at the image, which you see right now, that I had to think about this quote from the Swiss philosopher Lucius Burckhardt: "Warum ist Landschaft schön?" Why is landscape beautiful? And I think that's also particularly interesting when we think about an industrial landscape and how maybe we as artists can materialize an industrial landscape in our work. In French, we have this term, desert vert, which means in English green desert, the title of the work in the exhibition. And another nice one is marée verte, which means green tide, whenever we've got these big deposits of green which land on the coastlines. And I think these words are already very, well, poetic or strange, because they show us that a landscape, an industrial landscape, a green industrial landscape, is a dead space, a dead in terms of biodiversity. The industrial green color is some sort of promising culture, but at the same time a cultural and biological monotone somehow. And the industrial dream, it goes, I think we have many installations, like the first one we've seen, like Nuage Vert, where we're trying to work on that wavelength, which is typically around 532 nanometers. So obviously the range of color we can perceive best, which is also, I think, the color which you sort of see on this image. Anyway, this is kind of my favorite image. If you go onto the internet, you find a lot of images of Lake Erie in full algae bloom. And this particular one is on the first day of spring in 2012 by one of the kind of rapid response satellite of NASA. So I thought I'd write to the man in question itself to ask him how this image, the scientist at NASA who took the image, to ask him about the image and why. Because if you look online, all these images always really show this kind of beautiful fluorescent kind of color, which is always very sensational and, you know, in a way quite stunning. So he wrote back, Heiko, he actually wrote back within 10 minutes. I like people who kind of really write back within 10 minutes of being asked from somebody. The color depends on so many variables, whether it's a sediment or phytoplankton or both. The source of the sediment is the species mixture of phytoplankton and others. Another factor could be the look angle. The satellite is looking straight down, more or less, and the pixels are 250 meters square. A human looking down at a very small area, perhaps looking out from an angle ashore or boat, at a larger area. So this is really the fascinating thing as well, vision and scale in science and in art. The question of scale in science, because you know, so you have to have that vision, a kind of mediated vision through science to experience a sort of materiality, if you want. Because if I stand with my feet in Erie Lake, and I can only experience a sort of muddy green color, obviously, but seen through the satellite, I get a kind of complete vision of a fluorescent catastrophe. And I think this applies to many phenomena nowadays. I think the most famous one, as maybe you all know, is the ozone hole. We have to send a satellite to outer space to capture a kind of phenomena which is global, to be able to bring it into a kind of comprehensible vision. And so I think, if you want, we could, for example, Jens, could you please have the next image, which is
yeah.
So this leads us a bit to maybe also the work we have done with Susie, which is not what you see right now, but there's I just wanted to show you other works of us to contextualize a bit this approach. I think for us it's always important to find these, let's say, social, ecological, relevant images to people. And because we have, as in art, also as in science, a certain limited access to these images, like as we just saw with maybe the lake or the entire lake. Or for example, in this case, this is another work which dates a bit back. Fleur de Lis is a kind of mini nuclear power plant we have realized underwater, which explodes every 15 minutes, I think, and builds up over the day into some sort of landscape. How to re-access the phenomena which is inaccessible, and of course, they are from us kind of realized in some sort of humorous, mini theatrical way, which is, more or less serious depending on the context we exhibited. We have been also censored, for example, in this case, to show this work. But I mean, how to access a landscape which is too big to us to visit. So that was maybe at least the starting point for not all of the discussions we had, of course, with Susie, but one of them, and which led also to the two works which directly come out of this residency, which are in the museum. And, if we can go to the next slide, which is toy emission, if I'm right.
Yeah.
So this is, again, another work which is in the museum, but before I come really the work which we realized in collaboration with Susie, again, the same phenomena. This is a work we've done in New York, where we kind of try to access a phenomena which is really not accessible for us, which is really the, in this case, not a kind of a hybrid landscape. It's really like a man-made landscape. It's the city of New York, and the pollution or the cars, in particular, one strain of biological kind of man-made strain species, a kind of Porsche Cayenne car, which was omnipresent at least in 2007. So we make it small as a performance, and this is also what we've tried to do with Susie together. We make a small airy lake, and maybe we come to the last slide. And in the museum, you see that that we had this, say, our humble attempt to make a small lake area. A small lake area, on the one hand, a kind of miniature fluorescent landscape, which kind of is trying within a model to be very close to these visions we have, which seems to be important to us of these ecological phenomena. And another work is really a kind of the landscape built up really from the tiniest particle, from the algae itself, which come out of the lake, a landscape out of petri dishes where we constitute, where we draw with Susie together the kind of lake with algae. And again, because I think what we see on a macro scale, the kind of monotone landscape we see on a macro scale, is of course also a working method which is inherent in the kind of working ways of a laboratory. Also here we have to select and the strain which is the strongest, and to create a kind of a pure culture. So as in the laboratory, as also in Art Grande Nature and bigger nature outside, we have to aim for a monoculture and to develop a kind of a successful colony, strong colony.
Well, thank you, Heiko. I think that is a good point to just head over directly to Susie, because, of course, the work conditions that you've united are not the typical ones for her. But I think you mentioned many aspects that bounce back directly to research. So how did you experience this encounter with the artist, and how does it feed into your own research? What are the parallels? And why do you engage in this interaction? Because you need to be motivated as well.
Oh, yeah. I would like to talk about this. Actually, thanks, Adam. 2 years ago, introduced me to Helen, started some conversation, and this year, Heiko contacted me to working on this algae project. So, what?
The mic, there is a mic.
It's not good. It's working? It's better now? Close? Okay. Better? Okay. So why my research is a microbial process. I capture different kinds of bugs, bacteria, yeast, fungus, and algae to produce valuable products. Biofuel is one of that, and the bioproducts. And so, so when I talk to Heiko and Helen, they both interested in algae. This is the initiator of the project. And for me, I probably first introduce what we are doing here at MSU in terms of flue gas sequestration, CO2 sequestration, and then I will talk about how I feel about this bridge program. Okay, so actually we are working a group of MSU faculties, Dr. Liu in Biosystems and Engineering, and Dr. Mitch Smith and Dr. Angela Wilson in Department of Chemistry, and our power plant staff support our DOE Department of Energy project to study the biological and chemical ways sequester flue gas. So, and we work with California company who designed a specific photoreactor we installed in our pilot plant. So So why we do this? Because, we need to sequester CO2. I know there's, like earth scientists and engineers, we really want to do something to solve problem, not just like politics, just talk about it. And we do believe that global warming is a big issue, and we want to do something, okay? So give you, as an engineer or scientist, we like to use numbers to talk about things, okay? So, but talk to artists, when I always think about how could I describe this. So probably I just let you think about MSU whole campus. The power plant supports the electricity all over the campus, okay? So if we want to sequester all the CO2 from our plant, we probably need to culture algae full size of the whole MSU campus. So for the scientists who don't know how big the MSU campus is, the biggest single campus in the States. So other university probably bigger, but they have several campuses together. But MSU single campus is #1, the biggest in the States. Okay, so you need a full size of this campus in order to sequester all the power plant CO2 from every day. Okay, obviously that's impossible. So our research is to reduce the size and save a lot of water for algae culture. Okay, that's also the first challenge for algae culture. You need huge land and a lot of water. The second one is you need a robust strain. Okay, we are talking about years of culturing, not just 1 day or 2 days. We're talking about day and day harvesting, right? So you need a robust strain and can culture the autoclaved, unsterilized condition. Okay, so we select the strain actually from Great Lake area. So we try to find the strong strain we can tolerate the harsh condition and grows faster, so can compete with others and dominate the culture. Okay, so this is the second challenge, and we are working on actually in the lab. We selected the strain for many years. So at the beginning, the Great Lake in the water system, you have multiple algae strain, no matter, include also cyanobacteria. But we select one strain called Chlorella sorokiniani, which is a green algae unicellular, and we studied the initial work in the lab with the sheet culture, and in the lab I have a still photoreactor, but it's not that big. That one is 4 liters, and that one is 2 liters. So we use the blue and red light, and then make the purple, right? You are artist, you know the light makes the purple. Okay, so the blue and red, because this is the light wavelengths algae absorb that and it reflects the green color, so you see the green. Okay. So, so in the lab, and we will select the strain. So another challenge for the process is you need to find a good utilization of the algae. You need to have the market. So our research is to try to find fully utilizes of the algae. Okay, algae has lots of nutrients in there: protein, carotenoids, fatty acid, and lipids. So you only use to produce biofuel may not be economically feasible. So this is another research area we are working on to fully utilize every component in the algae. Okay, can you turn another slide? So this is we're talking about a team of us installed an enclosure inside of our power plant. Okay, this is a reactor designed by faculty. This helic coil, and this is the initial inoculation color. In a couple of days, you will see the green. It's emerald green or Spartan green. Okay, so it's a grows faster strain, and we're culturing this for more than 7 months. Right? So we take care of this, but we need to add nutrients. And this nutrient Dana mentioned about the waste utilization. Actually, nutrients can come from the waste stream. Okay, not only just you add fertilizer in it, you can get the nutrients from the waste stream and the water from what? From the power plant itself. Every day, you don't know, more than half of the water on campus actually utilized by the power plant. It's utilize a lot of processing water, the boiler and the purification process. So we use this water directly to culture algae. And so in this system, we can harvest every day from 30%, 40%, 50%, and 70%. That means this reactor is 100 liter every day till 70 liter we can harvest. The next, we put the fresh media water in there. The next day it come back. Okay, this is a fast growth strain. Okay, next. Oh, sorry. So, like I said, I like use the numbers to talk about things and make me comfortable to explain things. So this is the figures. Okay, you use these figures to describe the stability. And we capture this in the power plant enclosure, directly pump the flue gas in there. And this is till August. Now we are continue running until now. So every day you harvest, you add the nutrients. So this is the numbers show you the system. And you have the fluctuation part that is sometimes no CO2 supply. And the algae all turn to green, turn yellow. But this strong algae kind of proved our select strain is strong. It come back when the CO2 come back. So I have this 3 slides and to show you our research. And I will see the utilization of the algae biomass. This is this algae rich in about 60% of protein. So our CO2 sequestration, I mean, even just this small reactor I mentioned, it cannot utilize, absorb all the CO2, right? So work with chemists in the chemistry department will do the amino acid amine CO2 chemical absorption. That is much faster than the biological CO2 absorption. So basically, we use the CO2 culture algae, use algae biomass, extract protein, convert protein in amino acid, use amino acid to absorb CO2. That can be significantly absorb a lot of CO2 and reuse the amino acid many times. So this, from that way, we significantly reduce the overall size of the reactor and efficiently absorb the CO2. So this is the current project we are working on. So Yan asked me how could I work with Heiko and Helen on this project. Because when I talk to scientists like Heiko, let me think about things, right? So I asked him, what's the meaning you take the picture of liquid rain, right? So it's just beautiful. Do you want to say something? And then I think about it. This is arts, right? So arts give you observation. So what is science of engineer tell you? Give you numbers, code numbers. But so how could you link this? So I think about this way. Probably from the education perspective, I mean, a lot of faculty members work on campus, work on this bridge project. So arts is a way, you know, I'm thinking the science knowledge, if could be conveyed through in the form of arts, and could be easier touch the people's heart, you know, because the sensibility of the arts, right? So that could be, you know, help me, you know, inspire students' interest. So I don't know how could, you know, as a teacher, I cannot say I like teaching so much, but our job, right? So we have to let the students learn the knowledge. So if I draw a little bit Spartan on the petri dish for the first day of our engineering 101, you know, Dr. Brad Marks in our department, you know, just let everybody, every faculty when they stand in the hallway, talk to the students, what's your research? So let the students know, each individual student, faculty in the department, what's their research topic, right? So I can show this to the student. If this tiny closeness could inspire the student interest, I think it's worthy, right? So this is the part. I mean, also the magic power of arts could help, get the interest to learn science. Okay, so I don't know how I probably just use this opportunity to thank Yan, Adam, and Heiko, all this museum faculty staff helped me, working on this together. I feel this is a very cool project. So if, for me, I have fun. I've even think about I could let my students in the lab to have a patchy dish algae drawing competition for fun, right? So this is for my part and why I work on with artists in this program. So think about so in short words, I was thinking, okay, so science use cool numbers and artists use warm color. So science is cool and art is warm or hot, right? So together we can be both cool and hot, right? Just kidding. Yeah.
I want to come to one of the aspects, at least linking what Heiko described as socially relevant images, so a kind of mesoscopic condension of micro and macro phenomena that you're working with, but that in order to be socially efficient needs to be a condensation, a kind of compression that is aesthetically working in some way or other. But when you actually transcribed transposed it into the materiality, what I understood is that the glowing effect from the Lake Erie is also a kind of symbiotic relationship that the chlorella has with other bacteria, so that you see it as a glowing image, correct? And that in order to create this phenomena in a kind of reduced miniature that we can display in the museum, that would have been dealing with issues such as contamination, maintenance, and so on. So the solution that you found is actually to have algae cultures and in petri dishes forming the shape of Lake Erie, and on the other hand, to replace the algae, fungi, or bacteria, you correct me if I'm wrong, symbiosis, with actually plastic pigment. So we are choosing between two disasters, right? Between plastic pollution, microplastic, and algae pollution, right? So it comes back to how do you translate it into a materiality which makes sense. So here you have also this algae represented by plastic. I mean, it's an interesting solution that you both found, right? Why does it come?
So,
questions to both of you, of course.
Yeah. Heiko, you want first?
Well, I mean, just two things about materiality, or a couple of things about materiality. What I liked when I was, as Susie already said, I guess there are many different ways, and laboratories all over the world working with algae and developing algae strains and selecting them. And Susie was telling me that there are also laboratories which have machines which kind of do that kind of process more efficient. What I kind of appreciated in the way she worked, or she explained me, she worked is a kind of bit more alchemistic way, in a way that you really literally, you take these, you collect these algae, for example, at Lake Erie, and then you do it really by hand in the laboratory. So it's a bit kind of like a really hands-on, as I said, nearly alchemistic process where you kind of select them yourself in the laboratory and kind of see which one grows good and which one doesn't grow good, which is also very close, I think, to the way actually I guess we all work in our DIY way, and that kind of very close, being so close to the material, being so close to the material which comes from the lake, kind of meant that there was also immediately that kind of interface to then, to make that into an image. And from our part, that opens many new, of course, possibilities and forms and other things and other, let's say, other landscapes we would like to grow. And even I think Susie as well has many other research areas like growing mushrooms, etc., fungi. And so I think that's really, for us, the door was really the door into a new kind of series or exploration of works, because this is the first outcome out of the residency where I can see that I appreciate that message, and it has led to that kind of way of working, and will hopefully lead to new other works and ways of working. And I think the plastic thing was really like, as you can see in the museum, I mean, often we treat also phenomena with very unconventional materials, and I think it's always a very efficient way, at least in kind of getting people's attention, is to inverse it and to really confront something ecological with a totally surrealistic and kind of non-reasonable kind of material, and in this case plastic particles, which kind of simulate actually the best. I actually shouldn't even talking about this. It's always really frustrating actually when the artist gives away the last detail. Jens, you've got a tendency to reveal all our tricks in front of the audience actually. We could have just pretended they are really glowing algae, but now everybody knows they're plastic particles. But we have to somehow find the most bizarre materials to really, come close to the phenomena. And also it's more interesting for the audience because there's another piece in the show, and which is also an emission of cars, and it's not made with emissions, but with a material. I don't know if everybody's seen the show, so we have also got red emissions, pink emissions in the show. And Jens might give away the secret as well in a minute, how they're done, because you have been making them, didn't you, Jens?
The opening is only this evening, so we will discover.
Okay.
Just, Susie, maybe a last word about how does this glowing actually comes together and how it's so difficult to obtain in this situation?
Obtain so from you mean obtain the algae growth inside the culture system? So this algae, if you put on the petri dish, it can last for the nutrients in the media. The algal media can last for a month. So until March, I think should be enough nutrients to keep like this. Probably grows so slow, not like the photoreactor in the power plant. Every day you need to add nutrients, you need to harvest. But this one, it can maintain the green color, for several months. And also, like the question you mentioned about the plastic, right? So I think everyone heard about a plastic called a biodegradable plastic, right? So, but I don't have currently use this biodegradable petri dish, but hopefully we will have this as available, in the future. I mean, that's why it's not currently, because of cost, right? So but this is also research, and also need everybody working on this. Yeah. Okay.
Well, thanks a lot we have to break because we are a little bit running out of time. I can see that there is now coffee, which is probably more than quantum coffee. Thank you, Heiko, for being with us, and we are getting back to the roundtable in 5 minutes. Thank you. Thanks, Heiko.
Okay, bye-bye.